Hello everyone, this is your teacher Mrs. Namira Sahib. I welcome all of you to this class for biology class 10. Here we are going to talk about chromosomes and genes. As we know that the chromosomes are fixed in number in a cell, they have a fixed shape in the members of species. These chromosomes contain many genes, and now the question arises that what is the composition of chromosomes and genes? So here we are going to talk about the composition of chromosomes, which will be regarded as, or it will clarify the formation or existence of genes. So when we talk about the chemical composition of chromosome, when it is chemical composition of chromosome, we see that the chemical material of chromosome is called chromatin. Okay, the chemical composition of chromosome is called chromatin. Chromatin means a basic nucleoprotein. Okay, chromatin is a basic nucleoprotein. This nucleoprotein is called deoxyribonucleoprotein. Okay, it is composed of DNA and histone complex, means this nucleoprotein, deoxyribonucleoprotein, is actually consisting of 40% of DNA and remaining 60% as histone proteins, which are keeping the DNA proteins, which are keeping the DNA or the genes in their fixed position, right? So the DNA, which is called deoxyribonucleic acid, is made up of billions of units, which are called deoxyribonucleotides. When we talk about nucleotide, it is made up of deoxyribose sugar, phosphoric acid, and nitrogenous bases. Okay. So what is DNA? As we know that the DNA and histone complex. DNA histone complex means DNA and histone proteins are making up the chromosomes. And what is DNA? DNA stands for deoxyribonucleic acid. It contains sugar, which is called deoxyribose sugar. It contains phosphoric acid, and the third thing which it does contain are the nitrogenous bases. Okay, so these three components are making up a single nucleotide, and there are millions of nucleotides, billions of units, which are making up DNA. The DNA of a chromosome exists as one very long double-stranded fiber, which is also known as duplex. Okay, so DNA is double-stranded. Double-stranded. It is duplex, helical in structure. Okay, and it is single long double-stranded fiber, right? It is single long double-stranded fiber. Okay, this DNA, the long DNA, entire DNA is actually coiled and super coiled to get fit into a chromosome. Okay, so this highly coiled double-stranded, a very long double-stranded duplex, which is present in helical structure, it is super coiled to get fit into a much smaller space. And if the strands of DNA from a single set of chromosome and nucleus were laid out in straight line, it would be more than 7 feet, that is 2 meters long. It is too long to fit into a cell. So this is the supercoiling of DNA, which is making it enable to get fit into a cell. Now, how coiling of this long DNA is actually possible in a very small chromosome and nucleus of the cell? If we gently disrupt a eukaryotic nucleus and examine the DNA with an electron microscope, we find that it is because the histone is positively charged while DNA is negatively charged. Okay, so as we have talked about, chromosomes are made up of the DNA, which is only the 40%, and histone proteins, which is the 60% of the total chromosome structure. So this is very important to note that histone is positively charged and DNA is negatively charged. Due to opposite charges, they attract each other, and the beads of histone and DNA are called nucleosomes. When the string of nucleosomes wrap up into highly coiled structures, means supercoiling occurs, and the supercoiled chromomers form chromosomes. Okay, so genes are actually the parts of DNA, right? So when we talk about a special part of DNA, we are talking about a special gene, and each gene is responsible for a single function, for producing a single special type of protein or a special structure, right? So to understand the nature and composition of genes, we must know about the structure of DNA, because genes are localized parts of DNA having coded information to synthesize a protein working as enzymes. Okay, we'll talk about this in the next lecture. Thank you.